Welcome to our exploration of antigen processing and presentation, a cornerstone of the immune system's defense strategy. Imagine your body as a fortress, constantly under threat from invaders like bacteria and viruses. The immune system acts as vigilant guards, always on the lookout for anything foreign. But how do these guards recognize what's friend and what's foe? That's where antigen processing and presentation come into play. In this video, we'll break down these complex processes, showing how your body identifies and responds to threats. Let's start by understanding what antigens are and why they matter so much in immunity. Antigen is any substance that can trigger an immune response. Think of antigens as unique ID badges carried by invaders like bacteria, viruses, or even abnormal cells. These badges are recognized by the immune system as foreign, prompting it to take action. Antigens can be proteins, polysaccharides, or even small molecules. The immune system's ability to distinguish between self and non-self antigens is crucial for protecting the body without attacking its own cell. Now let's see how the body processes these antigens to prepare for a targeted immune response. Antigen processing is the first step in preparing an immune response. It involves breaking down complex antigens into smaller fragments that can be recognized by immune cells. Specialized cells known as antigen-presenting cells (APCs) are responsible for this task. These cells engulf pathogens, digest them, and extract the relevant antigenic pieces. This process ensures that only the most important parts of the invader are displayed to the immune system, maximizing the efficiency of the response. Next, we'll look at the main types of antigen-presenting cells involved in this process. Antigen-presenting cells, or APCs, are the immune system's scouts. The main types include dendritic cells, macrophages, and B cells. Dendritic cells are especially efficient at capturing antigens and alerting other immune cells. Macrophages, known for engulfing pathogens, also process and present antigens. B cells, while primarily known for producing antibodies, can also present antigens to helper T cell. These APCs play a crucial role in bridging the innate and adaptive immune responses, ensuring that the right cells are activated to fight off invaders. There are two main pathways for antigen processing: the endogenous and exogenous pathways. The endogenous pathway handles antigens from within the cell, such as viral proteins produced during infection. The exogenous pathway deals with antigens that come from outside the cell, like bacteria or toxins. Each pathway uses different mechanisms and leads to the activation of different types of T cells. Understanding these pathways is key to grasping how the immune system tailors its response to various threats. Endogenous pathway processes antigens that originate inside the cell. For example, when a cell is infected by a virus, viral proteins are produced within the cell. These proteins are broken down into small peptides by cellular machinery called proteasomes. The resulting peptides are then transported into the endoplasmic reticulum, where they bind to MHC class I molecules. This complex is then displayed on the cell surface, signaling to cytotoxic T cells that the cell is infected and needs to be destroyed. Exogenous pathway deals with antigens that come from outside the cell, such as bacteria or toxins. Antigen-presenting cells engulf these external invaders through a process called phagocytosis. The engulfed material is broken down in specialized compartments called endosomes. The resulting peptides are loaded onto MHC class II molecules and transported to the cell surface. This presentation is crucial for activating helper T cells, which coordinate the broader immune response. Major histocompatibility complex, or MHC, is a set of molecules found on cell surfaces that are essential for antigen presentation. MHC molecules come in two main classes, class I and class II. MHC class I molecules present endogenous antigens to cytotoxic T cells, while MHC class II molecules present exogenous antigens to helper T cells. The diversity of MHC molecules in the population is one reason why organ transplants can be challenging, as mismatches can trigger immune rejection. MHC class I molecules are found on almost all nucleated cells in the body. Their main job is to present peptides derived from proteins made inside the cell, such as those from viruses. When cytotoxic T cells recognize a foreign peptide on MHC class I, they are triggered to destroy the infected cell. This mechanism is vital for controlling viral infections and eliminating cancerous cells. The specificity of this system helps ensure that only abnormal cells are targeted. MHC class II molecules are primarily found on antigen-presenting cells like dendritic cells, macrophages, and B cells. They specialize in presenting peptides from external sources such as bacteria or toxins. When helper T cells recognize these peptides, they become activated and help orchestrate the immune response, including stimulating B cells to produce antibodies and activating other immune cells. This pathway is essential for defending against extracellular pathogens. Proteasomes are protein complexes within cells that play a key role in the endogenous pathway. They break down proteins into smaller peptides, which can then be loaded onto MHC class I molecules. This process is highly regulated to ensure that only the right peptides are presented. Without proteasomes, cells would struggle to process and present antigens efficiently, compromising the immune response to internal threats like viruses. The exogenous pathway, endosomes and lysosomes, are specialized compartments inside antigen-presenting cells. After engulfing a pathogen, the cell uses endosomes and lysosomes to break down the invader into peptides. These compartments contain enzymes that digest proteins, making it possible to generate the right fragments for MHC class II presentation. This step is crucial for activating helper T cells and mounting an effective immune response. Once peptides are generated, they must be loaded onto MHC molecules for presentation. In the endogenous pathway, this occurs in the endoplasmic reticulum, where peptides bind to MHC class I in the exogenous pathway. Peptides bind to MHC class II in specialized vesicles. This loading process is tightly controlled to ensure that only appropriate peptides are displayed, preventing accidental activation of the immune system against self-protein. After peptides are loaded onto MHC molecules, the complex is transported to the cell surface. This step is essential for displaying the antigen to T cells. The journey involves movement through the cell's internal transport system, ensuring the MHC peptide complex reaches the membrane intact. Once on the surface, the complex is ready to interact with T cells, setting the stage for immune activation. T cells are the immune system's specialized soldiers, trained to recognize specific antigens presented by MHC molecules. Cytotoxic T cells scan for antigens on MHC class I, while helper T cells focus on MHC class II. When a T cell receptor matches the presented peptide, the T cell becomes activated. This recognition is the trigger for a cascade of immune responses, from killing infected cells to coordinating antibody production. Cytotoxic T cells recognize a foreign peptide on MHC class I, they become activated and release molecules that kill the infected cell. This targeted destruction is vital for controlling viral infections and preventing the spread of abnormal cells. The precision of cytotoxic T cells ensures that only infected or abnormal cells are eliminated, preserving healthy tissue. Helper T cells are activated when they recognize peptides on MHC class II molecules. Once activated, they release signaling molecules called cytokines, which help coordinate the immune response. Helper T cells stimulate B cells to produce antibodies, activate macrophages, and recruit other immune cells to the site of infection. Their role is crucial for mounting a strong and effective defense against pathogens. B cells are unique among antigen-presenting cells because they can both present antigens and produce antibodies. When activated by helper T cells, B cells differentiate into plasma cells that secrete large amounts of antibodies. These antibodies bind to antigens, marking them for destruction by other immune cells. B cells also retain a memory of past infections, enabling a faster response if the same pathogen returns. Antigen-presenting cells, especially dendritic cells, can perform cross-presentation. This means they can present exogenous antigens on MHC class I molecules, a process usually reserved for endogenous antigens. Cross-presentation is important for initiating immune responses against certain viruses and tumors that do not directly infect antigen-presenting cells. It ensures that cytotoxic T cells can be activated even when the source of antigen is external. Vaccines work by introducing harmless forms of antigens into the body, training the immune system to recognize and respond to real threats. Antigen-presenting cells process these vaccine antigens and present them to T cells, leading to the development of immunological memory. This memory enables the immune system to mount a rapid and effective response if exposed to the actual pathogen in the future. The immune system must distinguish between self and non-self antigens to avoid attacking the body's own tissues. This process, called tolerance, is established during immune cell development. When tolerance fails, the immune system may mistakenly target self-antigens, leading to autoimmune diseases. Understanding antigen processing and presentation helps researchers develop therapies to restore tolerance and treat autoimmunity. Organ transplants introduce foreign antigens into the recipient's body. The immune system recognizes these antigens as non-self, which can lead to rejection of the transplanted organ. Immunosuppressive drugs are used to dampen the immune response and promote tolerance. Advances in understanding antigen processing and presentation have improved transplant outcomes by enabling better matching and management of immune reactions. Pathogens have evolved ways to evade antigen processing and presentation. For example, certain viruses can block the transport of peptides into the endoplasmic reticulum, preventing their display on MHC class I molecules. Others may interfere with the function of antigen-presenting cells. These evasion strategies allow pathogens to persist in the body and complicate the immune response, highlighting the ongoing arms race between host and invader. Cytokines are signaling molecules released by immune cells that influence antigen processing and presentation. They can enhance the ability of antigen-presenting cells to capture and process antigens, as well as stimulate T cell activation. Cytokines play a central role in coordinating the immune response, ensuring that the right cells are activated at the right time. Not all peptides generated from an antigen are equally likely to be presented on MHC molecules. The immune system tends to focus on certain dominant peptides, a phenomenon known as immunodominance. This selective presentation shapes the immune response and can influence the effectiveness of vaccines and therapies. Researchers study immunodominance to design better immunizations and treatments. Cancer cells often produce abnormal proteins that can serve as antigens. However, tumors may develop mechanisms to reduce antigen processing or presentation, helping them evade immune detection. Immunotherapies aim to boost the immune system's ability to recognize and attack cancer cells by enhancing antigen presentation or blocking tumor evasion strategies. Allergies occur when the immune system overreacts to harmless antigens, such as pollen or food proteins. Antigen-presenting cells process these allergens and present them to helper T cells, which then stimulate an inappropriate immune response. Understanding how antigens are processed in allergies helps researchers develop treatments to reduce allergic reactions and improve quality of life. Genes encoding MHC molecules are highly variable among individuals. This genetic diversity ensures that populations can respond to a wide range of pathogens. However, it also means that some people may be more susceptible to certain diseases or have different responses to vaccines. Studying MHC variation helps scientists understand disease susceptibility and improve personalized medicine. During infections, the efficiency of antigen processing and presentation can determine the outcome of the disease. A robust response can clear the infection, while evasion by the pathogen can lead to chronic illness. Researchers study these processes to develop better diagnostics, treatments, and preventive strategies for infectious diseases. As we age, the efficiency of antigen processing and presentation can decline. This contributes to weaker immune responses and increased susceptibility to infections and cancer in older adults. Understanding these age-related changes helps researchers develop strategies to boost immunity and improve health in the aging population. Therapies for autoimmune diseases often target antigen processing and presentation pathways. By modulating these processes, it's possible to reduce the immune system's attack on self-tissues. Advances in this area have led to new treatments that help restore balance and prevent tissue damage in autoimmune conditions. Immunotherapies harness the power of the immune system to fight diseases like cancer and chronic infections. Many of these therapies work by enhancing antigen processing and presentation, making it easier for immune cells to recognize and attack abnormal cells. Ongoing research is focused on improving these strategies for better patient outcomes. Chronic diseases like HIV and hepatitis can persist in the body by interfering with antigen processing and presentation. These pathogens may hide from the immune system or exhaust immune cells, making it difficult to clear the infection. Understanding these mechanisms is key to developing new treatments for chronic diseases. During pregnancy, the immune system must tolerate the fetus, which expresses antigens from both parents. Specialized mechanisms regulate antigen processing and presentation to prevent the mother's immune system from attacking the fetus. Research in this area helps improve outcomes for both mothers and babies. Inflammation is a key part of the immune response, and antigen processing plays a central role. During inflammation, antigen-presenting cells are recruited to the site of infection or injury, where they process and present antigens to T cells. This amplifies the immune response and helps eliminate the cause of inflammation. Immunodeficiency disorders can impair antigen processing and presentation, leaving individuals vulnerable to infections. These conditions may be inherited or acquired, and understanding the underlying defects helps guide treatment. Advances in gene therapy and immunology offer hope for restoring immune function in affected individuals. Parasitic infections pose unique challenges for antigen processing and presentation. Parasites often have complex life cycles and can evade immune detection by altering their antigens. Research into how the immune system processes parasitic antigens is helping to develop new vaccines and therapies for these difficult-to-treat infections. Viral infections rely heavily on the endogenous pathway of antigen processing. Viruses hijack the host cell's machinery to produce their proteins, which are then processed and presented on MHC class I molecules. This alerts cytotoxic T cells to the infection, enabling targeted destruction of infected cells and control of the virus. Bacterial infections often trigger the exogenous pathway, as bacteria are typically external to host cells. Antigen-presenting cells engulf bacteria, process their proteins, and present them on MHC class II molecules. This activates helper T cells and leads to a coordinated attack involving multiple arms of the immune system. Fungal pathogens can be challenging for the immune system, as they may have unique antigens and cell wall components. Antigen-presenting cells process these fungal antigens and present them to T cells, initiating an immune response. Research into fungal antigen processing is helping to develop better diagnostics and treatments for fungal diseases. Immunological memory is the ability of the immune system to remember past encounters with antigens. After initial exposure, memory T and B cells are formed, allowing for a faster and stronger response upon re-exposure. Effective antigen processing and presentation are essential for establishing and maintaining this memory, which is the basis for long-lasting immunity. Immune surveillance is the process by which the immune system constantly monitors the body for abnormal cells, such as those infected by viruses or transformed into cancer. Antigen processing and presentation are central to this surveillance, enabling early detection and elimination of potential threats before they cause harm. Immune system must be carefully regulated to prevent overreaction or underreaction. Antigen processing and presentation are involved in both activating and suppressing immune responses. Regulatory T cells help maintain balance by recognizing antigens and suppressing excessive immune activity, protecting the body from damage. Recent advances in technology have revolutionized our understanding of antigen processing and presentation. Techniques like mass spectrometry, advanced imaging, and genetic engineering allow researchers to study these processes in unprecedented detail. These innovations are paving the way for new vaccines, therapies, and diagnostic tools. Antigen processing and presentation are fundamental to the immune system's ability to protect the body from disease. From recognizing pathogens to developing vaccines and therapies, these processes are at the heart of immunology. As research continues, we can expect new breakthroughs that will improve health and combat a wide range of diseases. Thank you for joining us on this journey through the fascinating world of antigen processing and presentation.